those kinds of things.
Okay. Thanks for the nice introduction. So in today's talk, I will be focusing on mostly after you generate the data from these assays, what we do with it, like most of the how the processing of the data, and there are several steps on it. So before I get into that, so we live in the era where the field of biology has drastically moved from more from descriptive to more of the predictive analysis, and from qualitative to quantitative analysis approaches. And so it's more of like we used to call that it's like one gene, one PhD, meaning you are spending like 5-6 years on one molecule or one gene. But now we are like measuring hundreds and thousands of these molecules in parallel and quantitatively. So it's more of like predictive analysis, where again another example would be like more, instead of looking at the diagnostic markers, we look at the prognostic markers. Means you are looking for prognostic markers so that it could be more predictive of if someone is going to get disease, rather than once someone has the disease and you start looking for whether they have it. So or kind of for the diagnosis. So for biological measurements, we need more of these computational models. We have high throughput assays. It could be gene expression, cytokine or protein expression, cell phenotyping. So these are called high throughput assays. And I'll start beginning with the experimental design, which is very, very crucial, and then the batch effects. Normalization, multivariate analysis, database, software packages. So I'll touch on these points during my talk. So what's the biological question? What we are trying to understand? And being in the Human Immune Monitoring Center, we would like to know and the individual differences in immune response. And per se here, we have these immune components that we are trying to measure. And these are the from the whole blood, when you extract the serum and look at the cytokine expression, cell phenotyping from PBMCs, and the transcript from the whole blood or PBMCs. So how they vary across individuals, that's what we are trying to know. And today my focus will be on cytokines. And I don't think that this audience need much of introduction for cytokines, so I'll quickly go over it. That cytokines are the immune modulating agents which are released by cells. They interact with receptors on the target immune cells, or they stimulate specific target cells. And in context of what we are trying to do here is understanding the relationship between the cytokine stimulation and aging. And the disease is characterized by the dysregulation of pro or anti or pro-inflammatory cytokines. So here we do the quantitative approach to study the cytokine in unstimulated and stimulated condition in immune cells. But it is still unclear how reflective they are in predicting the immune status at cellular and organ level. So the assay that we have been doing here is the Luminex. It's a multi-bead assay where you have color-coded microspheres, and you have a blend of 2 fluorescent dyes, which we can come up with 100 different combinations, and each bead can have an address for a cytokine. And then these microspheres have the tags, and then you have a primary antibody which binds to the cytokine, and then you have a secondary antibody which has fluorescently labeled. So when you excite these microspheres which are going through this fluidic system, and then you excite it by the laser, one a green and a red. So if you say one laser, it excites the molecular tags, the other one is getting the address for the microsphere with which cytokine these are. So this is the basic concept of the Luminex. So now let's move on and talk about the experimental design and batch effects. So before even starting an assay, I will highly recommend that please consult a statistician and for an experimental design, because it needs to be compatible with what you are doing downstream data analysis. Because quite often I see that people coming and they say we did the assays, and if you see here there are 2 types of experimental design, right here, one on the left and the right. And if you see here, these are in greens are the biological groups, like it's, for example, disease, and the biological group 2 is a control. So in this experiment, you see it's a mix of, these are run in batches, so they are a mix of controls and disease and like so on, right? And on the other side, if you see here in the batches, in one plate you have all the controls, in the other plate you have another the disease, and then there's a mix. So can someone tell me which one is a better design? Okay, yes. So what I mean by can be corrected and cannot be corrected is if you have batch effects, you're running these in batches, right? So these are still, you can correct it because you have some samples in common here, which will we will be able to estimate the batch effects. In this case, since all the group, like all the controls in one plate or all the disease are in one plate, you cannot see the batch effects. So for a reference, you need something where it's consistently in all the 3 plates or 3 batches, right? And so the batch effects are contributing most of the times through the laboratory conditions, reagent lots, and personal differences, right? So the ideal design would be if you have you run the plates with reference samples, a common, some kind of a control where you can estimate these batch effects here. So this is what I would suggest, that before you run any assay, run by, look for whether you are compatible with all this data analysis and all the other effects that we see when we run the assays. So the typical workflow, once you get the assays, like you have the raw data from the assay and then from the Luminex system, and then we go through this rigorous QC, then a lot of filtering, the data which is low quality, we filter it, and I'm sure that you're going to go over these where we have assay checks which are detecting the systematic error in the assay and how we plot the CV standard curves. So I'm not, in the interest of time, I'm not touching those points at this time, but she will go over those. And then, you know, we have spent quite some time how to normalize the data. That's another challenge for these kind of high throughput assays. And then more of the statistical testing and data visualization. So starting with QC and pre-processing, the very first question that we ask, and those who are familiar with the Luminex, that we have standard curves. The question that we ask is, how reliable are the standard curves for estimating the concentration values for each cytokine, and then MFI or concentration value, which one we should go for, and then the precision in estimating the concentration values across plates or projects, and then we also did some of the cross-platform comparison as well. So now I will go over, and before I show you some analysis, I just want to remind you that, you know, how we play the game of dart, and we try to hit the bullseye, right? So just, this is an analogy where, you know, you have your measurements and you run in, say, triplicates, and you get 3 different values, and which are so far apart. So we would say this as a low accuracy, low precision. Here, the data, like the measurements are pretty close, but they are not accurate. So you will say these are precise. In this case, you can see that there is a bias. This bias can be adjusted. And then here, they are precise, they are not accurate. And this is the case scenario where you have more, like the data is accurate. It's, so high accuracy and high precision. So basically accuracy and precision, these are nothing but it's like you're looking at, the words are very similar, though they are different. And that accuracy indicates the proximity of the measurement, and the precision is measure of the repeatability or the reproducibility. And all these things needs to be, you should keep in mind when you are doing the data analysis. So in short, you can correct for bias, but you cannot correct for imprecision. So now moving on to for these high throughput assays or any kind of assays, we try to estimate how much is the background noise. And if you see here that These on the x-axis are all the cytokines. So in the Luminex system that we have right now is a 51-plex. That means you can measure 51 cytokines simultaneously. So on x-axis you have all the cytokines, and on the y-axis are the MFIs, which are the median fluorescent intensity. That's the unit for measurement. And so if you see here that all the cytokines, the background varies a lot. So it's not the background is similar for all the cytokines. Some cytokines have, for some reason, they show a higher background, some kind of a cross reaction, or some background estimates are not so consistent. So you need to keep in mind when you're doing the data analysis. So what we did is we did a small exercise. We are looking at if we adjust the background, and if we don't, then what's the difference? So if you see here, again, these are more on the concentration. So we measure from Luminex assay the median fluorescence intensity and the concentration. So the graph I'm showing you right now are based on the concentration values of these cytokines, and these are the MFIs. And if you see here that as with the higher concentration, you minimize the background effects. So at higher MFIs, you see low effects of the background subtraction, whereas when you see at the lower end, it's further apart, because blue is where the background was subtracted, and the red ones are where the background was not subtracted. So there's a lot of difference at the lower end. In the higher end, you don't. So so we reached to a conclusion here that maybe the background adjustment sometimes increases the variation at the lower intensity range. The other thing that we looked at is the standard curves. So being in a core facility, and we have lots of studies on Luminex. So the other exercise we did was kind of collect all these Luminex studies from different projects, and then we plotted all the standard curves. And if you see here, these are the colors are like different projects, and then we wanted to find out that how reliable the standard curves are, and it's turned out to be that the standard curves are pretty consistent. So that's a good indication, because when you want to compare across the projects, if the standard curves vary a lot, then that could be a problem, right? So then we also looked at the cytokine concentrations. Now if you see here, let me walk you through this plot where you have standard concentration, meaning we know that in each well what was put in. So these are based on the analysis on standards, where you look at the standard concentration of the cytokines. We did from 1 to about 5,000 picograms per ml, and these are the MFIs. So the red ones are the ones which are the MFI values. As I mentioned before, that MFI and concentration, these are the two units that we get from these assays. And so when we looked at the MFIs, and these are all like cytokines, these are all 50 cytokines that we plotted here, and at different concentrations of these cytokines, right? And then the one in the cyan is the ones which are the concentration. Oh, I'm sorry. I just said the other way, sorry. The red ones are concentrations, and these ones are the MFIs. And what we observed, that when you looked at the variation, you see most of the variation, these are the CVs. So you see most of the variations again at the lower end, but at the linear range, it's pretty stable. So this is your linear range, right? Where whether it's pretty it's linear, and the CVs or the variations are very low. But at the sigmoidal region of this curve, you see very high variability. So the problem arises when from the concentration values, these are from standard, right? And then you are trying to get an unknown sample where you're trying to get the estimate the concentration. If the values are around this range, you're okay. But if the values are here, they won't be estimated very well. There will be a lot of variability. So that was the reason that for this kind of the limitation for standard curves is that it does very well at linear range, but on the higher and the lower ends, there are a lot of noise.
Are you contrast to what Yael was saying, it looks like your CVs are higher on MFI than on concentration. Is that true?
No, I'm not going to show you these ones. No,
It
because
looks
it's
like
a.
more spread.
Well, if you see the MFIs
Right?
here, but we do the
I'm
MFI
just
info.
wondering, because this data would suggest that you are compressing the variation plate to plate by doing the calculation to concentration, and Yael argued just the opposite.
Well, these are concentration values. When we do with the MFI values, so concentration values you don't do normalize, but with MFI values we do the normalization, because here you are just getting straight from this standard curve. You are estimating how much the concentration is from the standard curves. But yeah, definitely we see here that here the CVs are higher.
Yeah, we should talk later about what's different between your two different
Okay.
data sets.
Okay, sure.
Yeah, just look at the opposite.
Okay. Then we also did some cross-platform comparisons. I'll just quickly go over it. We looked at the millipore and Panomics and different plates, and some of these the data is pretty reliable across the platform. It's not a much change. Though within the platform you see less variability, across platform you might see a little bit more. Now the concentration values are comparable across the platform. That's what we have observed for MSDs and Luminex, but not for all. For some cytokines we do, and in MSD some of the cytokines tend to perform better than Luminex. This is for for the researchers. Let's say that they started out with Luminex and then went to MSD. So what I mean here is that you can still compare the data. The other thing is we observed that log transformation it's a good approach when you are working with MFI values. So log transfer tend to do a more it fits the parametric assumption, right? So when you are down the line doing the t-test or any ANOVA or any other statistical test, it tends to do well. So always I would recommend that you do log transform the data. Then plate 2, plate variability, again the same message. It increases at lower intensity ranges. If you look at this, these are the cytokines. So some of the cytokines we see are pretty high in normal patients, and so here the variation is less. But again, most of the cytokines you see are from low to mid-range. There you see higher variability. So low abundant cytokines, you see more variations compared to the ones where you have a higher expression of the cytokines. So again, we are looking with where we have high throughput assays, multiple samples. How do you look at them in one plot? So this is an example where if you want to look at the reproducibility or the biological variation or outlier detection, this is called a scatter plot matrix, where you have these are all the sample names and then pairwise correlations. So you are looking at pairwise correlations for all these 14 samples in one plot. So this is a nice way of nice visualization where you can say which samples didn't work or have a very low Pearson's correlation, or they are outliers. So this is a suggestion for those who would like to see in this kind of plots.
It makes a lot of sense if you have the same control in multiple batches, for example, and you want to see which batches did the control perform identically versus like the two batches there where you can see
Right.
a lot of variability.
Mm-hmm. Mm-hmm.
Just for an example.
Right.
Can you explain a little bit more?
Okay.
What is the x-axis? What is the y-axis on here?
Okay. So these are all the MFI values. So we took the intensity values. Say, for example, you have two samples. So you want to look for, this is a correlation. So you look at pairwise. So this is a sample. One sample. This is another sample. You're trying to correlate all the cytokines, the MFI values of all cytokines.
So on the y-axis you have MFI of
Right. So here the y-axis is this sample, sample 2, for example, and the x-axis is sample 1. And the MFI values are being plotted.
For a given site?
Yes.
This is an array for a given.
Right, right.
Oh, I thought it was.
Oh, these are all cytokines,
All cytokines.
not one, all cytokines.
That's why there's 50 points.
Yes.
But each point is a cytokine.
Each point is a cytokine. So a cytokine measurement in sample 1 and what's the cytokine measurement for sample 2, you correlate it. That's what it's showing. But then you have 50, right? It's not 1, 50. That's why we have 50 data points.
Just to interrupt for a second, Angelica tells me, I didn't realize, but we actually have to be out of the room at 12. So I'm sorry.
Okay, we will be done,
Just to give you a
hopefully.
little warning.
Yes. Okay, so I'll move fast. So now coming to normalization. So we did a small normalization experiment where we had 16 normal subjects. I know I hesitate to say normal subjects because normal so-called, we just say healthy normals. But you know, in the lifespan, we all have we have different kinds of infections and, you know, right? So it's hard to say normal, but still assume that they don't have diseases, right? So there's 16 normal subjects, and they were ran in 3 batches on 3 different 51-plex kits. So these are like 3 batches, and each batch is a different kit. So so what do we see here? But before I get into this, we did some normalization techniques applied, and these are geometric mean scaling, intensity dependent lowest normalization, spatial z-score. For the interest of time, I'll just show you 2 of these. So what I'm showing here is a geometric mean based normalization. So what we do here is we have a lot of these cytokine expression values for normal healthy individuals. So we extracted all this info for about 90 individuals where we have kind of estimated the normal range of these 50 cytokines. And then, as I mentioned before, that the same set of 16 individuals were ran in 3 batches. So what you see here is that batch 1 in red, batch 2 in blue, batch 3 in green, and then we have a control. And when I say control, that means this is a geometric mean of 90 individuals, cytokine expression of 90 individuals. So it's a good estimate of a kind of a gold standard to which we want to normalize. So if you see here, these are before normalization for 1 patient. These are all on the x-axis are all the cytokines, and expression I mean here is the MFI values. And there's a batch effect. The green and reds and blues, these are for same cytokines, are moving up and down. And the black one, the dotted ones, are for the control, which is my gold standard here. And when I normalize it to that, if you see here that it helps, because there's a batch effect, right? And these are different kits. And we have seen that there's a substantial batch effect there. So what we should be normalizing to, for some time, we had been kind of debating what we should use. So then, since we have that information from these healthy individuals, we thought that should be a good standard to which we should be normalizing. So this is based on, instead of arithmetic mean, you use a geometric mean, because if you have magnitude differences in the batch, geometric mean corrects for it. The lowest normalization is another way of normalizing the data, because the geometric mean is mostly, it says one size fits all, or kind of scaling all the values by some number, right? With the lowest normalization, what it does is it's an intensity based normalization where it corrects. So if you see here at the lower end, again, this is the expression value, this is the MFI values, and these are technical replicates. So when they are technical replicates, we need to, like it should lie around 0, right? But it doesn't. It's a lot of, you see variations here. And so it's when you normalize it, then you see that it's pretty, like, you know, the variation goes away. This biology, she's asking me to go out, so I'm just kind of doing it as fast as possible. Multivariate analysis, let me skip this. I'll go over some of the visualizations we have. So instead of like typical box plots, you can also do some bean plots where you can look at the distributions of, say, in elderly you have a men and women, right? So you can have the distribution of both the groups in. So this is like more of a, in addition to just in the box plot, you don't get this information of the distributions. In bean plots you can. We did estimate the normal, the cytokine expression range in normal healthy individuals. That's what you see here. These are mostly density plots. I'll skip this. You can try the hierarchical clustering. That's another unsupervised way of looking at samples. And when you have like large number of samples, this visualization really helps you more of exploratory analysis. And what I'm showing here is that there are high leptin value levels in females. So if you see here, these are all patients. And then these are all cytokines, and I clustered it. And clearly I see a group where you see a higher leptin levels compared to the males. So these are all the females, there are some males. So this is kind of, looking at the patterns. And then another technique will be PCA. Okay. Sorry.
Sorry. So,
Yeah.
when you come back from lunch, please go straight to the rooms for the small groups. And basically, you'll segregate yourself into 3 groups. Like the first 10 people in each room will be the group that starts there.